All right. Hi, everyone. So welcome to today's Bat Advent Calendar. So I'm Dan Becker. I'm currently a postdoctoral researcher at Indiana University. Um, and this August, I'll be starting a faculty job at University of Oklahoma. Um, and so I'm a disease ecologist generally, and I focus on bats and their pathogens. So today, I'm just going to talk a bit about this paper that we published in Molecular Ecology back in April this year. So this was work in collaboration with a lot of other researchers. I especially want to kind of shout out to some of the early career people on this paper, including Dr. Kelly Spear, who's now at the Smithsonian, and Lexi Brown, who is now at Stony Brook doing her PhD. And so in this paper, kind of taking a step back, we were really interested in sort of the ecological and the evolutionary factors that determine, in some ways, cross-species transmission. And so there's been a lot of work, especially on viruses, thinking about what are the factors that determine how much infection a particular species has, and then how likely are they to share pathogens with other species. And we know that both of those processes, infection prevalence in that one host and transmission to a new host, can be shaped by sort of both ecological traits and evolutionary history. Most of what we know about sort of this cross-species transmission process, though, tends to be grounded in studies of viruses, because we tend to care about viruses more for human health and wildlife health and domestic animal health. But that also means that we have kind of a limited perspective on how these things operate in other pathogens. And in particular, bacterial pathogens actually tend to account for more zoonoses than viruses in particular. And we know that some bacterial pathogens like Mycoplasma gallisepticum, this poultry pathogen that jumped into house finches, caused really dramatic population declines. And so there can be these really big conservation impacts of bacterial pathogens themselves. This is all probably pretty salient in bats. And so there's been a lot of attention to bats and their viruses. We tend to know very much less about how bacteria spread within bat populations, what their zoonotic potential is, and whether these bats have negative impacts on bat health themselves. And so I've liked studying bat bacterial pathogens that sort of encompass both sort of public health interest and a conservation interest. And so in this paper, we studied a really diverse bat community in Belize, where myself and colleagues have been studying these bats for quite some time. We tend to catch around 30 to 35, at least, different bat species per year. And so we focused on sort of this high ecological diversity of this bat community to ask questions about how these ecological traits, how evolutionary history end up shaping infection prevalence and pathogen sharing. And we did that by focusing on this group of pathogens called hemoplasmas. These tend to be highly prevalent in wildlife. They tend to be fairly specialized. We think they're spread through a mix of sort of close contact and vector-borne transmission. And they can cause sort of acute and chronic anemia in their host, especially immunocompromised animals. And so there's, again, some potential here for sort of zoonotic impacts and also negative health effects on bats. And so we sampled this bat community, 2 different field sites across 2 different years. And so I think, sampled something like 400 individual bats. We sequence confirmed all of our positives. So sort of what do we find? So we found that generally sort of the heavier bat species tended to have a higher prevalence of infection. And so that might be due to sort of these interesting patterns between body mass and host competence. So larger species sometimes tend to be more competent. So that might sort of play a role here. If you're a larger bat species, you might be able to better generate new infections. But some of the more interesting stuff we looked at was at sort of the pathogen genetics level. And so we sequence confirmed all of our positives. We built a phylogeny out of them, and we classified infections into, I think, 24, 20, 28, I don't know, different genotypes. And so these aren't quite species, but we're calling them, at least genetically distinct types of hemoplasmas. And then we looked at how individuals varied in sort of, sharing these genotypes between species. And so some of what we found was sort of, kind of expected. So we think that hemoplasmas are generally pretty specialized, and we found that the majority of hemoplasma genotypes were pretty specialized on their hosts. We did see some cases, though, of genotypes being shared between bat species, and that tended to occur primarily kind of within clades. And so we found that, like you often see with viruses, sort of pathogen sharing declines with phylogenetic distance between hosts. And so if you are, Pteranodus fulvus, you're more likely to share with Pteranodus mesoamericanus. If you are one of the several Archibius or Dermanura species that we sample, you're more likely to share hemoplasma genotypes with those other species, which again is kind of expected. What was kind of neat, though, is we found a couple examples of cases where pathogen sharing was occurring between fairly unrelated bat families. And so we had one case where vampire bats, Desmodus rotundus, were sharing genotypes with Pteranodus fulvus, so more mopid. And then another case where we had Glossophagus orsina, Biostomid, sharing a genotype with Psychopteryx, another emballoneurid. So here, a case where we have, again, fairly unrelated species. But what's especially interesting about these sort of outlier cases is that there is some ecological context here for cross-species transmission. So in one of our sites in Belize, we have Glossophaga co-roosting with Psychopteryx. And so we can't really say anything about whether genotype sharing here represents contemporary cross-species transmission or a historic host shift. But it kind of opens the door for thinking more about how sort of interactions both within and between species might structure pathogen sharing. And so we're thinking about some future work in terms of, can we capture sort of these interspecies interactions, and will that sort of tell us something more about pathogen sharing, not just for hemoplasmas, but for other bacteria and for other viruses. Another kind of avenue this paper opens up is that we can start to think about if there is an impact of these pathogens on host health, or if they're zoonotic. And so in the vampire bats here, we've been doing marker capture for about 5-6 years now. So we're hoping to start looking, now that we have a nice data set, do we see any health impacts of having hemoplasmas as a whole, or of having different genotypes on kind of long-term host health. So in general, this was a really fun study to do. It's a lot of hard work of sampling bats, taking blood samples, and then having our collaborators do the pathogen screening. But I think we sort of learned some interesting things about sort of how ecological variation of this really diverse bat community manifests in sort of different patterns of infection prevalence across species, and different patterns of cross-species transmission. So if you have any questions, feel free to shoot me an email. The paper is open access on molecular ecology, so feel free to check it out. All right, thanks.